Hello guys, so we have done chapter number 7 till now, and in that we have learned about flower. So this is in this video we are going to learn practically about a flower. This is a hibiscus plant. As you can see, this is the flower. This is the stage between the bud and the flower, and this here we have a bud. Okay, so I'll be explaining you all of this. Also, with an example, I am telling you about the leaves. You can see here. So about the leaf, you can see this is the leaf, this is the back portion of the leaf, right? So we are going to use some tools. So firstly, this is the needle, okay? And this is the forceps. Basically, any forceps that you have to use for biological purpose, you will see this end is, the tip is very much pointed. Only the pointed tips can be used for dissection purposes in biology. So we have two forceps and one needle. So I am going to take all these instruments to show you all the dissections, right? So right now this is the leaf, you can see, right? So this portion, this central rib is the midrib, right? And all these veins on the side, these, right, and these, they are all reticulate venation, right? See these one, they are all tangled to one another, right? And they are not forming a specific network, right? So it is a very messed form of network. So this is a reticulate venation. In case of parallel venation, it would have been every vein will start from this point and end at the apex, right? Okay, so now coming on to the flower. So firstly we will dissect the flower, and then we will go to the to this stage, and then onto the bud. So starting with the flower, you see these are the sepals, right? And these are the petals. So sepals are these green structures you see, right? You can see in this stage also, these are the sepals, right? And in the bud stage, what you see, the sepals are covering the petals inside. You cannot see petals here. You can just see the sepals covering everything, every other part of the flower inside, right? So uh, in this now we will be dissecting. So firstly I am separating this. Now here we have put different stages of flower. So this is the initial stage, this is the middle stage, and this is the final stage, right? So firstly we are going to dissect this. So we'll keep these aside, and this. So you see these are the sepals, right? We are going to make a little cut here, and you'll have to be very careful while doing this, because you don't want to uh, disrupt the parts of the flower that you want to observe. Now see, we are going to separate these sepals. I am going to bring out. See, this is a crown shaped structure, right? And this is just covering the flower, no other part is there in between. So this is just basically covering the flower from the outside, right? So we are going to turn it around. This way, and separating it very with a lot of patience. And see, this is the, these are the sepals. This have been a little cut from the center, right? So this is a sepal from inside. right? This is basically a crown shaped structure. So we'll keep it at one side. Next is these petals, right? So now we will separate all of these petals one by one. You see the petals here are red in color. Yes. We'll have to do it very patiently. Now you see I have separated all the petals. These are the sepals, right? So this we have removed all the outer parts. Now we are coming on to the inner parts. Now you see here what we see. Firstly, we are going to separate one of the androgens. Now you see this part we have removed from lower part of this, right? So this is the stamen. Okay, I'll tell you what is the difference. Firstly, you see here this pink colored thin strand to which this little thing is attached. This is called filament, right? And this top portion, this is the anther. You can see this is bilobed. You see this structure. So this is the filament and this is the anther. This is a bilobed structure. I've already told you the structure. So this anther, this very tiny portion, this thing, this contains pollens, right? I'll try to break it for you. So we have separated filament and pollens. You can see this side red colored filament is present and this side it is the only the anther, right? So I've kept this filament. Now this is the anther. Now we are going to take our needle. We are basically scratching it now, right? Why? Because find we need to show you the pollen grains. See here. See these very tiny structures inside this. These are all pollen grains. I have cut it inside out. So these inside, these very small structures, these are the pollen grains.